I don't understand how you're always beating me. We've been training together for 2 years now, and you're still better than I am.
I don't know, Sheila. Maybe you just have to train harder. Actually, Sheila, you're training perfectly hard. You said you've been training just as hard as Megan for how many years?
2 years.
Yeah. Well, I'm reading this book right now. It's called The Sports Gene, and it talks about the study called Heritage, actually done by one of the researchers at Pennington Medical Center.
No way.
And they talk about how trainability is often characterized by your genes.
Huh.
Look, the book's right here. Do you want to borrow it?
Yes, I would love it.
Awesome.
Thanks. Remember the other day when you told me I had to train harder? Yeah, well, it's not my fault. It's my parents' fault. I just read this book, and let me tell you about the study that I found.
Jim Ryan wanted to be good at a sport, but most of all, he wanted a Letterman's jacket. Unfortunately, by middle school, he had tried a number of sports and had never become the star of the team, or even a member of the team. However, there was one sport he hadn't yet tried: track. At high school tryouts, he ran the 200-meter dash in just under 60 seconds, which wasn't a bad start, but wasn't good enough. However, he had briefly led the pack during the race, and that was good enough to motivate him to continue. Jim continued training for the team and ran his first mile at a conservative 5 minutes and 38 seconds, making him 14th on the high school team. 2 months later, he would lead his high school varsity team to the Kansas State Championship. Ryan continued training, his eyes set on running 1 mile under 4 minutes, a feat he would eventually conquer before joining the 1964 Olympic team. Interestingly, one teammate and friend of Ryan's began running at the same time as he did and completed all the same workout plans, yet never became an Olympian. Phenomena like these have led researchers to look to genetics for some indication of an individual's trainability. In 1992, under the direction of Dr. Claude Bouchard, 5 universities began recruiting 2-generation familial studies to participate in the Heritage Project. Heritage stands for Health Risk Factors, Exercise Training, and Genetics. The acronym lends some indication to what questions scientists were trying to answer. To what extent is trainability predetermined by genetic makeup? To execute this study, participants were given the same training plan to follow. Scientists found that some people's VO2 levels, an indication of endurance, increased drastically throughout the study, while other subjects' levels did not increase at all. The researchers in the Heritage Project found that the improvement of subjects was relatively consistent within families. As a result of the study, participating researchers concluded that 21 gene variants account for 50% of an individual's potential for aerobic improvement. In their experiment, subjects with 19 favorable versions of the 21 gene variants improved their maximum VO2 levels by nearly 3 times more than subjects who had fewer than 10 favorable genes.
Hey Sheila, thanks for letting me borrow this book. It was so interesting. Yeah, I did some more research, and genes account for a lot more than we realize. They even account for your BMI and how much fat cells are in your body by regulating this hormone called leptin. It's so cool.
In a study done in 2016 by Kilpalainen et al., researchers attempted to discover a genetic component to differences in blood leptin levels in humans. Leptin is a hormone that is produced by white adipose tissue or fat cells. Leptin circulates in the blood and can activate the central nervous system by binding to receptors in the hypothalamus. Leptin is responsible for long-term regulation of energy balance and is known as the starvation hormone. When the body has enough storage to perform normal functions, the adipose tissue secrete leptin, which signals to the brain that food intake is not necessary. Leptin is capable of suppressing appetite once this baseline of energy storage has been reached. Lower leptin levels are seen after short periods of starvation, and higher leptin levels are seen after high levels of glucose intake. Obesity is associated with the body's decrease in sensitivity to leptin, and genetic defects that cause a deficiency of leptin can cause severe obesity. Interestingly, leptin levels in the blood can correlate to a person's BMI and body fat percentage. People with a higher BMI and body fat percentage tend to have higher levels of leptin in the blood. It is believed that 30 to 50% of circulating leptin levels may be attributable to a genetic component. Therefore, certain people may be more likely to have a higher BMI and overall adiposity due to their genetics. This study sought to identify the loci associated with circulating leptin levels. They performed a meta-analysis on the results of 23 different genomic studies, including the Heritage study, and looked at associations between leptin levels in the blood and genotyped single nucleotide polymorphisms. The study identified 4 separate loci that influenced leptin levels that were independent of the amount of adipose tissue in the body. In order to confirm that these 4 loci were in fact associated with leptin levels, the researchers then performed knockdown studies in mice where these genes associated with leptin levels were effectively silenced using RNA interference techniques. This research into the genetic basis of leptin production may have consequences on the way obesity is treated in the future.
So I was doing my own research too, and there's this new computer program called Depict that geneticists use to figure out where genes are located and their specific functions too.
Oh, it's so interesting. Can you tell me more about that?
Of course.
Although the Heritage Research Study made significant progress for a genetic link to endurance abilities and benefits reaped from its physical activity, it was found to be very difficult to pinpoint genes to the pathways through which they were acting. Thus, a computational tool was developed to predict the functions of specific genes, identify pathways, and pinpoint areas of high gene expression. Depict, or the Data-Driven Expression Prioritized Integration for Complex Traits, identifies, enumerates, and assigns the characteristics of each gene set to a specific membership probability. It can then match the gene in question to a gene set based on the probability based on its characteristics and how well they match with that of the gene set. If the probability calculated by Depict is greater than it occurring by chance, it will then group together those genes with similar functions into associated loci and identify tissue types with likely high gene expression. The primary advantages to this tool is its use of gene set prioritization methods, as well as its ability to be independent of phenotype-specific hypotheses. The results of using Depict often come in the form of producing a hypothesis that can be used in genome-wide association studies like Heritage, all in an effort to better match genes with their specific location and function. Currently, more work is being done to expand the gene set information available to Depict, specifically with respect to cell and tissue types, as well as incorporating epigenetic factors into the tool.